All right. You are good to take it over whenever you are ready, Carol.
Yes, perfect. Roman, you're okay. I'll introduce you first. Hi, thank you for attending the PCA Computational Seminar Series number 2. I'm the chair of the committee of the Plant Cell Atlas, Computational AI and Modeling Committee. My name is Carol Huang. I'm a faculty member at New York University. Today is my great pleasure to introduce my colleague at NYU, Professor Romain Lopez. Romain is an assistant professor joined with computer science and biology at NYU. He received his master's in applied mathematics from École Polytechnique and his PhD in electrical engineering and computer science from UC Berkeley, advised by Professors Michael I. Jordan and Neil Youssef. He was a postdoc at Genentech and Stanford University, hosted by Avi Ragas and Jonathan Pritchard. He has received Best Paper honors at leading AI and computational biology venues, as well as the Stat Wonderkind Award, which recognized North America's most promising early career scientists. His research develops probabilistic and causal machine learning methods to uncover mechanisms that govern cellular behavior in disease, including the SCVI framework, which I'm sure a lot of you have used, and the SCVI tools ecosystem for deep generative modeling and single-cell omics data. So without further ado, Roman, please take the microphone. And
Thank you very much. Is my sound going through okay?
yes, but we
Wonderful.
cannot see you.
Yes, yes. So I will. You cannot see me? Yes. Okay, that should work now.
Yes, great. We can see you
Great.
now.
Okay. So, hi everybody. It's a great pleasure to be here. You will notice that I have put also my plant background. So I must say that so far I didn't get much chance to work on plant biology, but I believe that a lot of the tools I will present today are actually of interest to the community. And I hope that feel free to reach out to me with any questions or comments or collaborations by email. I will share my slides now. So. All right. Are my slides going through okay?
Yes, very well.
Wonderful. Okay, so today I will talk about, my talk will focus on a specific technology, and throughout this technological example, I will also talk through computational methods for analyzing this kind of data from regulatory genomics. And so here I will be talking about genetic perturbations and single cell measurements. And this specific story will be focused around the merging of two technologies, so profiling those perturbed cells through imaging and through sequencing, RNA sequencing. And this is work that I've done during my postdoc, and that is coming out soon, but on bioRxiv. And right now I'm professor at NYU Current Institute in Computer Science and NYU Biology. So and please, I don't know if this is the format of this talk, but there is enough time, so I don't mind being interrupted with clarifying questions throughout the talk. Thank you. So for the purpose of this talk, the technology is very usable, but I will focus on macrophages because that's what we tested the technology on. So we all know macrophages central role in the innate immune system, and one of the most pictorial thing that we can remember about macrophages is that they're able to they have this phagocytic activity, and so they can attack and digest bacteria. And so we can see here through a time lapse what's going on. So beyond this, what's very interesting about macrophages is that even if we know them mostly for their phagocytic activity, we also know that they have very multifaceted function, and they're actually involved also in disease. So, for example, macrophages have an inflammatory activity, but they also promote tissue growth, tissue repair, tissue surveillance. And so basically, when some of the balance of these three type of phenotypes or categories of phenotypes go wrong, we can see inflammation, and here those specific disease I might mention if we get through the end of the talk. For example, in cancer, we also know that macrophages are very involved there because they disrupt this balance of function. So whenever we have function like this, it's usually a good case to kind of understand what are the genes that regulate these functions. Okay, and so CRISPR has been an amazing tool for running screens, and those screens are helpful for discovering those genes. Okay, those regulators. So here is an example of phagocytic activity screen from Michael Bastik's lab, where basically macrophages were cultured and then made to, in a pooled library. So there is a genome-wide CRISPR library. All the macrophages there have been perturbed, and we're basically able to positively select by making those macrophages eat beads, and then with the magnet, we can then separate these macrophages and then basically do sequencing. And when we do this, we do a screen where we look for regulator of phagocytosis. So this is what the data looks like. I reprocessed the data. I plotted it here for the purpose of the talk. And so we can see that basically we have those genes with negative effects, some of the actin genes, and it looks like the actins are kind of necessary for phagocytic activity. If you break those genes, you don't see much phagocytosis. And on the right, you can nominate genes that are, if you knock them out, you will have a positive effect on phagocytosis, which is kind of interesting, but I will not dig into that. So the problem with those screens that we may all use and we're familiar with is that somehow their measurements remain univariate. Univariate meaning there is a single outcome to those perturbations. And it's a very powerful tool, but it's kind of a problem. So we can run, for example, one screen for phagocytosis. We can run another screen for inflammation. If we were able to stain for a protein, for example, that is a marker for that, we can do another screen for antigen presentation. And we can think of many others, but somehow we need to do a new experiment for each readout. Also, we have a problem of interpretability, which is that there can be different molecular mechanisms that converge into the same phenotype. And if we just get a list of regulators, we're not actually able to deeply understand all those pathway converge into a phenotype, which, for the purpose of finding mechanism is very important. And finally, what if we want to care about a phenotype that we don't even understand well, so we don't have markers for? And so in that case, being less biased would be really important. So there are two technologies that are important for this purpose. The first one is Perturb-seq. In Perturb-seq, we're able to basically take this pool of cells and profile them with single-cell RNA sequencing. And there is an important trick that consists in being able to read out the guide, the perturbation as well. And once we do this, we get two matrices. After processing, we get two kind of matrices. The first one tells us which gene is expressed in which cell, and that's a regular output from single-cell RNA-seq. The second one is a guide capture matrix that basically tells us which gene has been perturbed in which cell. Okay, and when we put this together, we're able to understand which genes are regulating expression of other genes, which is a very powerful framework. Another kind of technology would consist in taking those same perturbed cells and putting them through a microscope. So here we get the similar challenge of we need to have two kind of measurements. The first one that tells us what is the phenotype of those cells, okay? And the second one, which is what was the actual perturbation in those cells. So the first one can be done with immunofluorescence staining techniques. And the second one can be done in many ways, but what I will discuss today or briefly mention is basically in situ sequencing, where we can literally one nucleotide at a time read out the guide or a barcode for the perturbation. And after doing this, we can basically have a computational pipeline that tells us that gives us some features about the morphology of the cells. Okay, and when we start comparing perturbed cells versus control cells, we're able to get a good sense of what has been changed in those images. All right, and what's important to understand to contrast those technologies is that the cost of Perturb-seq is basically pertains to the number of cells you will profile, because you need a minimal number of cells per experiment. And that can be costly. For OPS, though, because imaging is much cheaper, as long as we're able to prepare and process enough plates, we're able to do many more perturbations. So that explains the x-axis of this plot. On the y-axis, it's pretty understandable that with Perturb-seq, you can measure tens of thousands of expressed genes if you have enough high enough quality of the data. But in OPS were bounded by the number of channels in the microscope in general. And so you imagine that you will have tens of markers maximum. And so the purpose of the talk today, or I guess the technological excuse I was mentioning, is that what could be very interesting is to try to think about combining the strength of those two technologies. And so this is a work that we did with my colleague Taka Kudo from Genentech. And so the way that this experiment I want to mention works is we basically profiled both imaging and sequencing on the same pool of cells. So we took the bone marrow-derived myeloid cells, we differentiate them into macrophages, and at the same time that we do that, we also induce CRISPR perturbations. And once the cells are stable, we stimulate them with lipopolysaccharide to observe the polarization towards inflammatory phenotype. And once we have this, after 9 hours, we basically run two experiments in parallel. One of them is with a small library, so with a library of 1,000 genes. I mean, I say small, but it's rather large, but with a non-genome-wide library, let's say, or like a medium-sized library, where we will look at so RNA sequencing and also a few surface proteins. And on another library, on another pool of cells, again, those are not the same cells, we can look at 3,000 genes knockout. In principle, you could do genome-wide here, but we were bounded by the labor time of the imaging. So now there are robots, so you can technically do genome-wide. We can look at the imaging. And so now we have this multimodal data set in which for all the perturbations, we know what are the changes in gene expression, what are the changes on surface proteins, and what I call here morphology. I will talk a little bit about what we actually image, but basically we can image proteins and also some what usually people would call morphology, so nuclear and other parts of the cytoplasm, actin filaments, etc. Okay, so this is experimental setup. I don't know if people are able to actually ask questions, but here would now be a good time to ask questions if there are any clarifying questions. Okay, I assume that either there are no questions or people cannot ask questions. So I will move on to the next slide.
I do not see any questions, so it's fine.
Nice, thank you. So what's great about this, or what's interesting about this data? Let's see, add something in the chat. Oh, I can't see the chat. Oh, can somebody read me if there is a question?
No, it's not a question. It's basically saying people can Q&A and the Q&A.
Wonderful, wonderful. Thank you.
Keep going.
Thank you. Appreciate it. So what can we do with this data? So first, it's a kind of an interesting use case. It's one of the very first use cases at scale where we can understand, do morphology and gene expression agree or disagree in how they capture perturbation effect, okay? Is there one technology better than the other? For what kind of biological questions? This is all unresolved and unknown. The other part that I'm a fan on, because I guess I'm a computational scientist, is can we use one technology as kind of an anchor or a substitute for the other? Okay, so if imaging kind of encapsulates the gene expression data really well, but is less interpretable, maybe we can predict one for the other, and I will try to go through this a little bit today. And then there is another interesting question, which we will see if we get to, which is all this screening data that happens in vitro can be not always relevant for macrophage biology or macrophage disease biology. But in this work, we've actually tried to relate the effect of perturbation to variation in population scale, so human genetics and whole exome sequencing.
So Roman, we have a quick
Yes.
question. About what the timing for the imaging and CySeq, are they done at the same time?
Yes, so this is a really good point. So it technically, right, those are different technologies, which mean that the best time point the best time point, I'm going to define what that means. The best time point could differ per technology, okay? And here, the best time point, for example, in the context of a perturbation screen would be, what is the time point at which we observe the most separation between the perturbations or the most perturbation effect? Or it can even be, what is the time point in which we observe the most significant differential expression in LPS response versus baseline? So obviously we haven't done enough time point screens to know the answer to variation I was mentioning. But what we know though, there are many, this LPS system has been very well studied in the context of the second variation, which is how many genes are differentially expressed in LPS at time t versus like non-LPS. And usually gene expression can peak around, I think it's 12 hours actually. And we know that imaging can be the most different. No, sorry. Gene expression is 6 hours, and imaging is more like 12 hours. And so what we did is kind of taking the middle point and assuming that it would be all right. The reason was that we wanted to have a common time point to be able to really make those interpretations of, this technology captures this well, this technology is captured that well. But at the end of the day, I agree that all of those, conclusions will be bound to a certain time point. And it could be that something you don't see in gene expression at a given time point, you could see it at a later or earlier time point. And it's a difficult thing to kind of get around. Thank you. Okay, so if there are no other questions, I will move. So this was the motivation for why this data set is interesting. And then I will first talk about the Perturb-seq, and I will talk a little bit about methods that I've been working towards for analyzing Perturb-seq data. And then I will talk about the imaging and how we put them together. So this is what the sequencing data looks like. So at the end of the day, we get around 300,000 cells. The number of genes we can capture with some decent qualities around 14,000 genes. And because we had a 1,000 gene perturbation, we had 4 guides for each gene, and so we get 4,000 guide RNAs. And the one thing I want to spend some time on is how do we actually analyze this data. So uh, the thing that was there from the very early days of those genetic screen was regression, okay? And intuitively, you basically want to be able to fit a regression and get coefficients of size perturbation by genes that tells you what is changed after the perturbation. So this is equivalent to calculating some kind of log4 change. And what you obtain, and this is from the first Perturb-seq papers, this heat map on the right, they knocked out transcription factors there. So you can see how each transcription factor so you can cluster those. So here those are called gene modules, and this cluster of modules or groups of transcription factors, and you can see how they affect on the columns, on the rows here, the programs. So those are expressed genes. So it's the effect of perturbation onto all the expressed genes. And you can see that, for example, that specific module here is a group of TFs that has down regulating effect on those genes, those group genes, okay? And you can see this really nice checkerboard pattern. So the advantage again is that it's easy to read, and here I'm showing you, so those transcription factors are actually JAK-STAT or STAT1, STAT2 here, and they upregulate, because it's a KO, so negative means upregulation, the program of antiviral response. And this is very well studied in those BMDMs and BMDCs. What's a bit limiting though is that they don't systematically account for single-cell heterogeneity, and they requires a lot of data processing and understanding before you actually get to a really nice heat map like this. What doesn't require usually as much processing is some of the work that I've been doing early on during my graduate school, which was around those latent variable models and those deep generative models. And the reason for which they don't work very well in this perturbation setup is that those methods or any other single cell analysis method is usually based on separating cell states and in kind of an unsupervised way. So those methods are meant to capture the dominant pattern in the data, and they never use as knowledge the labels which perturbation was in which cell. So because of that, if you apply a CVI to the data, for example, all the cells we profile in the experiment I'm talking about, you can see, so there are some kind of clusters, so there is probably some interesting biology happening here. But basically, there is a high separation of the cell cycle. And let's try to dig into this to understand what it means. It's normal that the cells, are cycling at some point, and it's normal that they're represented here. But what's kind of that cluster space on the right, you would end up, separating, for example, this cluster and separating that, thinking that there are different perturbation response, for example. And actually what's happening is that in all of these clusters, for example, in that cluster here, you can see the cells polarizing with one side of the cluster is this whole cluster is basically the same perturbation, this M6A writers, but you have this polarization according to the cell cycle. And it's the same thing on below here. But again, the problem is that because this latent space spends so much parameters trying to explain cell cycle, which we don't really care about. We know that some of the cells will be cycling at the end of the day, but what we really care about here is which perturbations have the same effect and which perturbations have a different effect, for example, and what is this effect? And so this is a case where the single cell heterogeneity can kind of hurt you if you don't interpret it in the right way. And so this is all happening because, again, SCVI has no knowledge, or any of those PCA and all of those methods have no knowledge of the perturbation identity. And so instead, we would like to change those methods to kind of exploit the perturbation label. So this is the leading question here. How do we extend the single cell analysis methods to capture the effect of perturbation? So there is a lot of work around this. I will here talk a little bit about basic principles, and then I will talk about a specific method. So the idea is that we can come up with causal models that actually write down this is what the effect of a perturbation look like. So instead of having a model that doesn't know what the perturbations do, we can have this idea of latent variable intervention, in which the little hammers here are kind of encapsulating the fact that perturbations can actually perturb in terms of so biological perturbation can be mathematically modeled by those little hammers, and they change the equations, etc. And basically the idea is that so perturbation will change some of the prior parameters for these latent variables. And let's go straight to the next slide. And this is actually not enough to think about it at this level, but what's been working really well and was proposed by so many people that I will not cite all of them, but the person who actually taught this kind of framework to me was Ethan Weinberger, when he came as an intern in my group at Genentech. And I've been working in this kind of models for quite some years now, where we basically want to separate the variants. So basically, some of the variation in the single cell data is the same across all perturbations, and we're going to factor that into the background, like the cell cycle, because it's not really what we care about when we care about perturbation effect. And we're going to try to segment the effect of perturbation in a different space that here I call the salient space. You can see the hammers are kind of acting only here and not on that part on the left. And so again, basically we have now, instead of having one PCA, if you want, or one less set of latent variables, or one SCVI, this new model will give you two spaces. The first one contains a variation present across all the control cells, and the second one will be the space where the perturbation actually have effect. And there is a lot of mathematical formalism, and it's a really interesting area also as a machine learning scientist. You can do, and I'm going to pass through this slide, you can also do, there is some beautiful mathematics coming out of this actually, but I'm going to pass this for today. And what's really nice is that it's also a generic tool, meaning that you can extend it to multimodal data. So for example, again, we have our background space or salient space, and you could apply it to, here it's gene expression and surface protein. You could apply it if you have an ataxic or if you have intracellular protein. You can basically extend the mathematical framework and just extend this model, which makes it very appealing and really nice for many new emerging assays in perturbation profiling. So now I can show you what the two spaces look like. So for example, in the background space, what we would like to see is a lot of the cell cycle variation. And we can see that here we do see cell cycle variation. And for example, if you knock out this checkpoint for the cell cycle, you can see that the cell cycle score is changing significantly. And the cell cycle score is actually computed directly from the latent space. And it hasn't, yeah, the model doesn't even know that it's cell cycle, but it's basically captured there, which is really nice to see. And what's really beautiful about this is that this in the background, there is not just cell cycle, there is other, trade-off in activation strategies. So for example, what we have found are two other important gene programs that appear in the background. One of them is a complement activation, and the other is lipid metabolism. And you can see that they have very different patterns. And you can see that actually it's different genes that are down-regulating one program and activating the other program. And you see this kind of trade-off like this. And so again, here we're in the background. So the control cells, they all have this gradient of complement activation and this gradient of lipid metabolism, but you're able to still find perturbation that kind of activate these switches, which is really interesting. So, and now I can talk about the main space that we're interested in, which is the effect of the perturbations. And so, I'm not comparing to the other, I guess, latent variable. We do this in the paper with metrics, etc. But the idea is that it gives you a really beautiful view map to look at because you see all these clusters, and these clusters are very relevant. So here, the colors are actually given from this. You take this single cell embedding, you pseudo bulk it. So on the left, every point is a cell, but on the right, every point is a gene. Okay, and so we cluster this pseudo bulk embedding here, and then we project it back onto this UMAP. And so you can see very beautiful localization of a lot of processes. So all of these cells here have a knockout that correspond to the M6A writer. I will mention this at some point. And like this, you can find again so the JAK-STAT or right here, TLR4 or right here. So whenever you knock out TLR4, basically the cells look unstimulated. And you see you have a very strong response here. And so this is what we get out of those perturbation modeling frameworks. We obtain this map for which perturbations are similar and which perturbations are different. And then with the same model, you can still go back to what people like to read, which are those heat maps. Here, these are the perturbation clusters, and here these are the gene expression programs. So like how the genes are expressed or co-regulated across all those perturbations. And again, we can find that JAK-STAT upregulate this interferon antiviral response. Okay, as was documented before. I just picked on that, but you can look at this heat map in a very unbiased way and try to make sense of it, etc. Are there any questions? I think I'm closing the Perturb-seq section here. Are there any questions on the Perturb-seq?
So I have a quick question about the salient space.
Yes,
Is
yes,
it
yes.
something that you, as an input to the method, or is it learned?
Yeah, no, so it is learned. So basically, the way that the model works here is that the model only needs to know there are two data sets, basically. One of the data set is a background data set, and one is called a target data set. So the background data set is all the unperturbed cells, and the
Okay.
target data sets are all the other cells. And then the background space will try to explain all the variants in the control cells. So control here means control guide. And then the variance that is not explained by the background will have to be explained by the salient
I
only
see.
for the perturbed cells. So that's why all the cell cycle and all this stuff will go in that space,
I see.
while
Okay.
the perturbation effect or like what is not, or like gene programs that are not represented in the background will have to emerge in the salient.
Okay, makes sense. Thank you.
Nice, thank you. So this is learned, yes. Okay, any other questions? Okay, so I will move towards the imaging now. So first, I think it's a bit of an unusual, imaging is still an imaging is still an emerging technology. So I will try to tell you a little bit more about what I mean by imaging. It needs to be a bit more precise. So I'm talking about this technology called optical push screen, again, in which we can profile which perturbation was in which cell. And we can also look for some immunofluorescent channels. And so, right, so first we will do, the way it works is once you have your pool of cells, first you do the phenotyping. I mean, the cells are fixed, of course. You do the phenotyping in several rounds. So for example, we can measure P65, we can measure NOS2 and IF1 alpha and SPP, osteopontin, SPP1. So a lot of macrophage markers or macrophage relevant markers. And now, and after all that, we need to use in vitro transcription. So it's a little bit of a technical point, but in general, you would have no more guide RNA after doing all this immunofluorescence and bleaching. And so to kind of revive the RNA, you need to use zombie transcription or in vitro transcription with the T7 polymerase. And this allows for then doing in situ sequencing. And this is what's called Perturb-view. So that's why I use the word Perturb-view here and not OPS because of this T7 polymerase in vitro transcription step. And after all that, so I think we total number of cells imaged was probably 20 million, and we lose half of them because of the fluorescence might not work that well and etc. But I think it's still a pretty good turnout, and we have around 1,000 cells per guide RNA basically, or a bit less, but almost. So what's really helpful here is that we can do feature extraction, and then we have this effect basically on the perturbation and to the imaging. So let's look at the specific, how, I'm going to say this one more time on how the imaging works. So there are multiple phenotyping rounds. In each round, we can measure a certain set of proteins with antibodies. And basically, you can see on the first round of antibody, P65, which stain the nuclei here, but actually P65 can translocate under some conditions. NOS2, which is a marker for inflammation in iNOS, and you can see it's in some of the cells, not some others. You can see the vimentin here really nicely. And we can bleach and keep going for having more markers. So at the end of all experiments, we have all of those protein stainings, which makes it one of the richest perturbation data sets, I believe. And it's not enough to do phenotyping. We also need to do, after in vitro transcription, in situ sequencing. And so those images down there are the same, then the same cells, and you can see in the upper view. And so this this is right here. So basically, after each we do by cycles, and you can read each one nucleotide per cycle, and you can conclude after enough cycles, so 20, I forgot what the number is in this data set, you can uniquely identify which gene was perturbed from your library. Okay. Any questions on the assay? Okay. So and we can, what we did first was to try to understand whether the screen worked because this is relatively novel data. So here we basically looked at the P65 brightness, so like total intensity, and you can try to understand when I target P65, do I have noticeably lower intensity? And these are randomly picked images. And so you can see, there is some overlap, and I guess CRISPR has some efficiency, right? Or there could be some mislabeling, but it's also due to CRISPR efficiency. Knockout can have escaper cells. These are all cells. But overall, there is a really strong effect, which is really nice. And we can do this for other markers, and we can see that, for example, so relay is a gene encoding P65. This is a maximal effect, and NOS2 has the effect for NOS2, AIF1 for IBA1, and et cetera, et cetera. So this was really nice to see. But we didn't do this screen just for univariate type of measurements, as I was mentioning. And so what we were able to do is, after embedding all the cells and massaging the latent space a little bit, so here this imaging is notoriously harder to work with. And I think my lab will maybe dedicate some time doing research in that area because there is a lot to do. We were able to get some embeddings, and this is what, again, each point here is a gene that's been perturbed, and the clusters correspond to genes with similar perturbation effect. And you can then, based on what are the name of the genes and their function, you can cluster and give meaning to those clusters. So, all right, we've made it. We've looked at imaging and sequencing, and now let's finally get into the weeds of talking about these modalities together. Any other questions before I get there? Okay. So the first thing we wanted to do was basically try to understand whether the similarities, so how two genes have a similar effect in one modality versus the other, whether that was consistent. Okay, so we basically concatenated the two maps, and we built a dendrogram. So here, the way that the genes are next to each other, this actually corresponds to somehow the joint effect, okay, the effect on both. But the color scheme here is a cosine similarity, so it's a measure of similarity between perturbations based only on the transcriptome, okay? And so you can see that, for example, all of those triangles here will correspond to groups of genes, and we can look at their function, and everything makes a lot of sense, okay? So the interferon genes are here, for example. All right, now we can complete this heat map by looking at the imaging. Okay, and what's pretty striking, right, is that a lot of those triangles are completed into squares, though not all of them. I just pointed at this one. We're going to talk through specific examples, but and if you look at statistical measures of similarity, those heat maps are overall very, very, very similar to the point that you would imagine that they're just consistent. Okay. But so the heat maps are mainly consistent, which means that those two experiments recapitulate a good amount of shared biology. Now we can start zooming in to look into the specifics. So what's really nice with this experiment is that here it's nice, you can see the name of the gene. So this is our JAK-STAT cluster. You can see that how two genes are similar in Perturb-seq, how two genes are similar in OPS. You can also see their effect on those expressed genes cluster, those gene programs, okay? And you can see their effect on some of the staining, okay? And these are the main or like most easily interpretable protein markers. So, or this is all good. So this mapping itself, I believe, is very important and very interesting. What we can do is zoom in further to try to and make sense of some of the biology. So, for example, this is a striking example of two groups of perturbation that look alike decently in imaging, but that look completely different in sequencing. Okay, so this is interesting. Those genes here are members of the M6A writer, and those part of the JAK-STAT complex. And so the way you can interpret this is that basically these are two kinds of perturbation that have the same effect, meaning that they basically inhibit those interferon signaling genes, but in different ways. So in one case, JAK-STAT is kind of basically needed for as a transcription factor for expression of those interferon signaling genes, but the M6A writer is kind of needed for the translation. And so if either way, the phenotype in terms of imaging, because imaging doesn't capture gene expression, they will look very similar. But when you look at the gene expression, basically, in one case, those genes will be expressed, and in one case, they will not. Okay, and so this is kind of an interesting anecdote. You can find quite a few like this, but what we were very surprised with is that actually PerturvView, so imaging is more sensitive. Okay, so the reason I'm saying this is that the heat map I've showed you with the concordant effect was only those 500 perturbations that appear to have strong effect in both assay, but actually we had 1,000 genes in commons, and out of these 1,000 genes, 300 have an effect only in imaging and not in sequencing. And very few actually have an effect only in sequencing and not in imaging. And that was very surprising to us. So what we wondered was, could this come from technical effect? Could this come from cell numbers? Could this come from many other things? It's unclear, but we have one clue that I can tell you about. What we did was taking those 300 genes and understanding where they are functionally related to each other, okay? And it turns out, like, for example, 90% of a certain cluster from the imaging, cluster number 5, these genes were they all had effect in imaging only. And there is some enrichment for 2 other clusters. So what do those clusters contain? Number 5 contains mTORC signaling and mitochondrial proteins. And cluster number 3 contains protein trafficking. And so what was really beautiful to see is that, for example, in the context of module 3 of protein trafficking, you can see accumulation of osteopontin. So basically, when you when you remove those this ability of the macrophages to secrete osteopontin, SPP1 is resecreted into the milieu. So basically, you obtain uh, uh, accumulation of this protein that maybe at 9 hours you don't see effect on gene expression, but you see that the imaging channel is completely different. And so that's why it's very easy to nominate this as a hit in one modality, but not in the other. And similarly, for this mTOR signaling, we see drastic changes in a phosphospecific antibody, which means that somehow, with imaging, we can have phosphospecific signal, and we cannot have that in sequencing, and so that might explain those differences. Any questions on this story?
So,
Okay.
quick question. Your
Yes.
imaging data, is there like morphology data part of it, or is it
So there are
just
unfortunately, morphology is a word that's been used rather unwisely, and I don't think I've done the best job at using it in the most rigorous way. So morphology sometimes can mean bright field imaging, sometimes can mean quantitative phase imaging, and sometimes it's been used also to designate, for example, cell painting, in which people stain for actin filaments, microtubules, and DAPI and other, like channels that stains for the organelles, for example, and people call that morphology. So in our experiment, we do have some of those minimal cell painting markers like actin filaments and DAPI, etc. But we do not actually have bright field imaging, for example. We don't have like structural feature of the cell in a sense. Yeah. And but the reason we added so many proteins is that it allows us to basically make this kind of interpretation that you couldn't if you just have I guess the if you just have those cell painting measurements. So as a conclusion, the similarity matrices of the perturbation effect seems to be overall very concordant. There is some level of discordance, and it looks like each assay can capture different aspects of the biology. So this is actually not surprising in a sense, but it's really nice we can characterize it this way. And what I haven't talked about is that if you, by doing some analysis of correlation, you can look at, for example, what are the gene programs that are systematically associated with some optical features like translocation of P65, for example, or accumulation of osteopontin. And so one thing that I'm thinking about working with my lab is trying to understand whether we can extend this kind of framework for correlation for understanding in a data-driven way what are convergence and divergence of how several gene programs can, for example, convert into the same optical feature effect. So there could be many mechanisms that lead to P65 translocation, and being able to nominate them with some full discovery rate would be very important. So this is the kind of stuff I'm interested in as a follow-ups to this work. But what is really nice is that this suggests that PerturvView has enough signal to predict Perturb-seq. And so for the interest of time, I'm gonna think about maybe Carol, should I try to finish at 12:50 so we have time for discussion, or what is the best way to use the remaining part of the time for the talk?
Yeah, maybe wrap up in maybe 10 minutes, and then we have about 5-minute questions because I wanted to see what you have.
Sounds good. Okay.
Yeah.
So, uh, so the idea is that now for many perturbations we have imaging, and for some of them we have sequencing. And there are two use cases I'm going to briefly discuss. So either we have, for some perturbation, we actually have sequencing, but we have so few cells that we can't exactly figure out what's going on. And so in that case, it would be interesting to think about merging the information we have from, we know the imaging looks like this. So, and we have a few cells, so we have some signal, it's just noisy, and we can denoise it by merging the data together. And obviously, a most important application would be, let's say we have no cells at all and we would like to impute. How is it limited? So these are the two setups, and we came up with this method called EB-MoCaV. EB is it's a bit of a mouthful name to the method, but EB means empirical base, and it's a statistical tool that's quintessential to somehow how this method works. So the idea is this method takes as input the genomics data. So genomics means not the imaging, okay? So it takes the genomics data, the single cell level, the gene expression, the protein, and what perturbation was in which cell. And it also takes a summary of the imaging data. Okay, this is very important. It's very hard to work with single cell images and single cell gene expression and try to merge that because the data, the modalities are very different. It's another topic of research for my lab potentially. But what you can do is you can summarize the images with those embeddings, and you can use the embeddings as a way to predict the model at the single cell level to predicting the effect of perturbation at the single cell level. So this is what we do here. And briefly, briefly, the way that this works is for every cell, we can use those modality-specific encoders for getting a sense of what does a background space look like and what does a salient space look like. So all of this is actually part of the model I was describing earlier. And these are variational distributions. So it's kind of like taking as input the data and then guessing what is a latent variable. And once you have that, you have the generative model on the right. So for the perturbed cell, as I was explaining, we will use both of the latent representation, background and salient, and we will put them through neural networks to guess what are the parameters of the distribution. So negative binomial for the gene expression and mixture of negative binomial for the protein abundance. But for the control cells, we will zero out the salient because for the control cells, we want them to be explainable only from the background. That's why it's called the background space. Okay, so this is the the MoCaV part that I was mentioning earlier, and to this we add the empirical base part. And in the empirical base, we basically have another neural network that takes as input the summary of the imaging and that tries to imagine what is the location in the background and in the salient of those perturbations. And so when you do this, you're able to then impute, you can denoise and you can impute the effect of perturbation for unseen perturbation. All right. And so this is where the imaging data comes in. Any questions on this mechanism? Okay. So basically, once we're here, we can overlay the predictions from the data. And we can see that things overlay decently and there is some specificity, so it's pretty nice to see. We were able to impute all the perturbations that we had seen only with imaging. And we can, again, as a way of looking at the data, we can zoom in into the specific cluster. And what's really nice is that you have examples like this, where, for example, in LAMTOR, so red is imputed and blue is observed. So LAMTOR3, for example, clusters well with all the LAMTOR components. The COG complex, so some of them were only in imaging and they all cluster next to each other. And here the commander complex clusters pretty nicely, where actually we had only this CCDC22 that was measured in both imaging and sequencing. And we're able to line up all the other perturbations next to it, for example, including so most of the complex here. And another interesting example is those SRP proteins. So there is this kind of nice cluster here of those proteins that are sharing this same name. And we can try to understand. So what's nice is that we don't just have a latent space here because we have a full single cell model. We can also understand to the level of the gene programs which genes are changed specifically in those perturbations. And we can do this even without having the gene expression for any of those, right? So those SRP, none of them were actually in the Perturb-seq. And we can nominate gene programs like this that are upregulated or downregulated. And what's very interesting is that what's downregulated is ER stress and protein homeostasis. And actually, this lines up really well with the known role of this protein that are part of this. The main pathway they work with is this regulation of aberrant protein production. So they're very important for proteostasis. And we have some validation on a Perturb-seq screen. So the last thing I want to tell you is about benchmarking. So showed you some kind of, a cherry-picked example where we can look into the latent space and we can see and we can make sense of things. And this is a nice story, but to be more unbiased, we need to do some benchmarking. So how to do it? There are two ways. One of them is purely computational, where we basically take some perturbations out and we pretend we've never profiled them, and then we see how well can we predict that. Okay, of course, everybody can do this. Another thing that is a bit more involved is to look at the predictions, to pick interesting predictions, and then to run another Perturb-seq data, another Perturb-seq experiment, and then measure the accuracy based on that. So that could be more dangerous because there are batch effects and other things, but we're going to show that it's still very informative. So again, we take this embedding, we cluster, we pick in what we call interesting perturbations with strong effects. We show that the correlation of the log-fold changes across the two experiments are pretty high. So we're happy that there are not too much batch effects are not terrible. Much better than we ever thought. And somehow then what we can do is compare, for example, different methods and trying to see their performance on how well they recapitulate perturbation effects at the level of individual genes. And what's very interesting is that there are many methods here. Some of them are, so the first two are based on imaging. So here lower is better. That means that those are the two best methods. And those other methods are based on gene pathway, LLM embeddings, and other techniques. And basically, what's coming out of this is that it doesn't really matter if you have a fancy model or not, though ours work a little bit better. But what really matters is the input. So OPS seems to be, or imaging seems to be a really good like input data for predicting the effect of perturbation, which justifies our paradigm of doing a larger library on imaging and a smaller library on sequencing to save sequencing cost, but still have the interpretability of the sequencing. So this is our test data set, so the perturbation that we did on the new round of Perturb-seq that have never been provided to the model. And of course, we use the same gene program clustering than we had before. And you can see this is a dendrogram and a heat map visualization this is the data, okay? And we're trying to predict that, but this is never fed into the models. This is what our pipeline predicted, okay? So if you squint with your eyes, it's interesting. It's not perfect, right? So I can see this stripe here, for example, as the wrong sign. But let me show you what the second best, but we know it's useful because we're able to understand the effect on those SRP proteins, and it predicted well at least some aspect of biology. But this is the second best baseline. A model, an AI model that doesn't use any knowledge about the imaging. And the idea is that imaging is a really powerful framework for at least getting a broad sense of what's going on in Perturb-seq data. Okay, and basically those models, I believe, are almost uninformative, like for this purpose that we were trying to achieve here. Any questions on the benchmarking, and then I will close my talk. Okay. So as a conclusion, with this machine learning model, we can impute and denoise Perturb-seq data. I haven't talked about denoising because it would take too long, but it's an interesting prospect. The imputations were interpretable, and they told us about what's going on with this SRP cluster. And I think it's pretty impressive that we're able to get an interpretable outcome for perturbation that we profile only through imaging. The OPS, again, is the most important contender or the best performing framework or like data for predicting the effect of perturbations, which tells us something about some maybe the field of perturbation prediction. For those of you who are familiar with it, and yes, and the denoising is interesting. So I do not have time today to go through that part. There is a recording of this talk probably online that will discuss that part. And for the interest of discussion, I will move to the conclusion. Up, 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 up. That was quick. We made it. And yes, I will just move on to the questions. Thank you very much.
Thank you, Roman. You have questions, please put it in the Q&A. And maybe I will start with a question before people can type in. I have a question about the first part, about the salient, um, the two-part, the base.
Yes, yes, yes.
Yeah, I forgot the
The
terminology.
background and salient space.
Oh, right, that
Yes.
space. So if we have cell type specific data, what would you recommend if we want to see cell type specific perturbation effects?
Yeah, yeah.
How
So
would you frame the analysis?
So there are, yeah, so this is kind of interesting. You can do a thought experiment to understand what would happen. So in principle, right, if you say that your background data set is unperturbed cells, then you would have all the variety of cell types would appear in the background space,
Right,
and then the effect of perturbation would be in the salient. Um,
right.
I don't know, but then what would be missing to me in terms of modeling is how do we capture interaction between
Yes.
cell type and perturbations? And so the problem is that because the model here is nonlinear, the interaction can, I mean, I guess they would go in, I guess, right? So you could just have perturbations separate in the perturbation space, cell
Mm-hmm.
types separate in the background space. And then
Yeah.
because the model is a neural net, it can kind of like do the nonlinear interaction by itself. But it's an
Mm.
interesting problem because what we care about is somehow, I mean, we do care about interpreting the interaction, but then that would be and this model is really nice for interpreting which part of the variation is in which space or the other.
Okay.
If you care about which gene programs are like affected in one cell type but not the other,
Yes, yes,
you will
yes.
need to interpret the model post hoc. So, for example, you need to
Post hoc.
query it with the differential expression and try to understand which genes are maximally different under,
Mm.
one cell type and the other. And then if you start working with PBMCs or like with there are many cell types, you get into combinatorial like pairs, like what does it mean exactly to have? I mean, that would be the same with linear model and interaction. But I think this kind of like cell type specificity, people have just studied it under like two cell types, and you can just say, which variance in the perturbation effect is shared and which one is different. But
Mm.
once you start figuring out like PBMCs or like larger set of cell types, it gets pretty difficult to interpret that. I think this is an kind of like one of the emerging problems as like there are many new perturbation data sets like this, and people want to figure out cell type specific regulatory networks. It's definitely very interesting.
Okay, good. Okay, I have a question from the Q&A. The question is, I was very intrigued by the fact that imaging seems more sensitive overall, with many imaging-only hits, but only a few RNA-only hits.
Yeah, me too.
Do you know what kind of genes those RNA-only perturbations correspond to? Are they enriched for specific functional categories or
Yeah.
regulatory processes?
Um, I should do this, and it's not the first time I'm asked this. I guess we'll have to do this during when we do the revision work, which will absolutely address this. I remember looking once because I was curious, and basically there was first like it's hard to do an enrichment of 16 things out of 16 clusters. Because I mean, actually there are 63 gene programs, like sub programs, and 14 big gene programs, and basically the 16 don't belong to a single program.
Oh,
Right, so that kind of spread around multiple programs, multiple modules, and so it wasn't clear for me. It wasn't obvious. Yeah, we need to look more, but it wasn't obvious. Yeah.
any more questions? Please type in the Q&A. Joanna, do you see more questions in the Q&A? I'm worried that maybe I'm not seeing them.
Yeah, if you actually, here, I can actually just copy it for you real quick.
Yeah, that would be great. It seems I'm not. Right, okay, I see it now. Next question. In cases where perturbation effects are cell type, cell state specific, maybe similar to what I asked earlier, for example, acting only during certain cell cycle stages, could separating cell cycle into background
I can read them.
space. Yeah, okay. You can see that?
Thank
Okay,
you. Yeah,
great.
yeah.
Yeah.
Yes, so I think we've kind of yes. Actually, I've worked with the student at some point in some interaction type latent models, but we haven't applied them to that specific type of data. So there is a whole semantic of of models like this, and technically you can add an interaction term. I just don't know. I just haven't applied it to that data enough to kind of understand whether that really addresses that specific problem. But potentially, yeah, you can extend the modeling. And one of the big problems is kind of like the non-linear identifiability of the model. And so you can address this under some conditions. It gets a bit technical. But in the case that we've evaluated here, I think for us, the cell cycle is more, it's not a nuisance, but it's just, we're not interested in some perturbation effect that are conditioned on certain cell cycles. For us, it's more the perturbation will push cells into different cell cycles. And so it's a little bit of a different semantic. And in the problem we've studied, what the person is asking, we haven't encountered exactly. So we need more investigation. It's a very interesting question. And I can ask
Last
the
question.
last question I can ask. Yeah, I can answer the last question. So, yes, the salient and background training, it's in a single, I don't understand what training cycle mean, but yeah, in the single optimization procedure, we solved for the salient and the background. Yes, it's the same, the same training. And then are there goals to include are there goals to include annotations for dosing and duration to allow measurement of that moderator heterogeneity? I'm not sure I completely understand the question. So dosing, in principle, you can add a lot of things in the semantic of the salient space. So you can have a dosage curve, for example, inside of the salient space, and you can try to model that. And you can also model like time. For example, we actually have some follow-up work where we have time points. But one kind of problem is that to have nice dosage curves, etc., you need to have many time points. You need to have many dosage points. If you just have very discrete conditions, like 3 dosages, 2 time points, it gets a little bit difficult to kind of interpret exactly what's going on. So I think this will be very data dependent. And it might pertain more to how do you apply the given method rather than how to extend it, because again, it's really hard to work in those middle regimes like this where you have only few dosages and few time points. Yeah. Thank you. Then there is a question. Have you applied your current method to plant samples? Absolutely. I have not so far, but it would be great. I think what is kind of interesting is that so I know a few of my colleagues are working on CRISPR screens, even at single cell for plants. And so that would be a great application case. But yeah, it's still ongoing, and I think the data sets are kind of emerging. But I hope that a lot of the machinery that exists for CRISPR in mammalian cells will be applied to plants and allow a lot of discovery and engineering of plants. Yeah.
Great. With that, I think we have a very nice closing for this webinar. Let's thank Roman again for a great seminar and answering the questions. Okay. I
Thank
don't
you
know
so
if I
much.
can see. Yeah. Yeah. So thank you very much, and we hope to see you in the next round of the PCA seminars.
Thank you, everyone. Also, I do just want to let everyone know that we will send out an email with recordings as well as a certificate of completion for everybody. So keep an eye out on your email for that. And with that, thank you, everyone. Have a great day.
Thanks, Roman. Thanks, Joanna.